Good afternoon, everyone, and welcome to the next in our series of the Connect on Coronavirus webinars. I'm Doug Brown. I'm the Chief Executive of the British Society for Immunology. Thank you very much for joining us this afternoon. Again, we have hundreds of people viewing this webinar live, and already those that have been recorded and put on our website, we've had thousands of people view them online as well. So it's fantastic to have this engagement, and not just from a UK audience, but from a global audience as well. So thank you very much for joining us. And thank you too for the British Society for Immunology's prestigious journals for helping make these webinars happen. Immunology and Clinical and Experimental Immunology have provided the support to get these webinars off the ground and accessible to all of you. It's my absolute pleasure today to be introducing two amazing people at the coalface of the COVID-19 research efforts at the moment. We have Professor Peter Openshaw, who is Professor of Experimental Medicine at the Imperial College London and past President of the British Society for Immunology. And we have Dr Ryan Thwaites, who is Research Associate at Imperial College London. And they are here to tell us about the work they've been doing within the ISARIC 4C consortium, which is a really exciting collaboration, pulling scientists together from all over the UK to answer quickly lots of questions that we have on COVID-19 and to make that data, make those insights openly available to the research community. So today we will hear about the initial findings from that collaboration, but also we'll hear their views on some of the questions that have been posed in a report that the British Society for Immunology launched in partnership with the Academy of Medical Sciences a couple of weeks ago on what we do and don't know about the immunology of COVID-19. So it will be a fantastic talk to hear about all of these things and new insights into COVID-19. As with the last presentations, the logistics, will have our speakers for talking for around about 30-35 minutes between the two of them, and then there will be time for Q&A at the end. So please do submit your questions via the question function on the webinar platform, and I'll ask as many of those questions as I can at the end of Peter and of Ryan. And do bear with me now as well, because we're about to test, well, at least for me, the first time, a new functionality on this webinar platform, which is polling. And so Peter and Ryan would really like to get your opinions on two key questions before and after the presentation. So get your fingertips ready to vote in these polls. And the first question is up, I believe, and it is, do you think you could be infected twice with SARS-CoV-2? Answers are yes, no, or maybe. So we'll give you some time. Excellent. So we've had 3/4 of people voting. We'll leave it there, and I will share the results of this one. So you can see the yeses win. Not many people think we can't be infected twice, and a bunch of you in the not sure category. So we'll log those scores and we'll come back to those at the end and see if you think differently at the end of the presentation. We then have poll number 2, which is, which statement do you agree with most about COVID-19? And the 4 choices are on the screen. Yeah, just over half. Excellent. We'll close that one and share the responses of this one. So the vast majority went for option 2, which is that it's mostly beneficial, but not entirely. So I will log those scores as well. And again, we will come back to those at the end. So thank you very much for interacting in that way. It's now time to hand over to Peter and Ryan, and we very much look forward to your presentation.
Well, great. Well, thank you so much, Doug, for that wonderful introduction. I think I would totally agree that Ryan is at the coalface. I think it would be generous to say I'm even at the pit head. I'm probably down in the governor's office more often. But anyway, I certainly am spending all my time on COVID-19 now, a virus that we didn't even know existed just a few months ago. So we thought we'd try and make this a bit more interactive rather than just having one person speaking for the whole time. And I thought doing a double act with Ryan would be great fun. So I'm going to do a bit of introduction and then hand over to Ryan to talk about some of the immunology. But during my introduction, I'm going to emphasize this wonderful ISARIC 4C study that I'm so proud to be part of, and which is being led by Kenny Bailey and by Callum Semple. I'm one of the co-leads, but really, they have come to the fore during this outbreak. So let's move on to the next slide. So this is a beautiful picture of the virus, which has just come out in the New England Journal. I like it particularly because it looks like a sort of snake coiled up inside this rather atypically completely spherical viral envelope. You can see those large spikes on the surface. This is really why coronaviruses are called coronaviruses, because they've got these spikes on the outside looking a bit like the crown, and the spike is interacting with the with ACE2 on the cell surface, and there's lots of great papers coming out showing the distribution of ACE2 and the way in which it's affected by things like allergy or by smoking or by age. So the distribution in the respiratory tract and elsewhere is being very well studied, and that really accounts for a lot of the tropism of the virus. Let's go to the next slide. You can see that coronaviruses, although they were first described back in the 1960s, and actually the UK played a major part in describing coronaviruses. David Tyrrell at the Common Cold Unit on Salisbury Plain was instrumental really in characterizing these viruses, and did some early studies where common cold coronaviruses were used as challenge agents in human infection volunteer studies. So coronavirus has got a very unusually large genome for an RNA virus, and because of that, they have unusual abilities to edit any mutations, but do have a pretty regular mutation rate, which allows people to go back in time and to see how long the virus has diverged from putative related viruses in other mammalian species. And many species, well, all species really that have been studied are the host of typical coronaviruses that sometimes can jump between species. So going back in time, maybe something like half a millennium that the human coronavirus NL63 first jumped species and came into being a human common cold virus, and then 120 years ago this OC43 virus jumped, and then the Hong Kong U1 virus probably in the 1950s. So they have this sort of molecular clock that allows you to study just how long they've evolved as a separate lineage, which gives some insight into their transmission between species. Okay, so I think a lot of us were first woken up to the importance of coronaviruses in 2002 when SARS emerged. I think this was very alarming. Those of us who are sitting on pandemic planning committees were terrified of this one. It looked like it could easily go global, had a very high mortality of about 10%, and it looked almost unstoppable once it had managed to get to Toronto, but it was just stopped in time because of severe lockdown. MERS, on the other hand, less transmissible but more lethal, so about 30% lethality, but fortunately hasn't really quite had the same potential to spread. So next slide shows a bit of the history of the emergence of this particular coronavirus. And this is a graphic which has come from a lovely publication in JAMA, which came out quite early in February of this year. So remarkable how fast it's been that the Chinese in particular have been very quick to share any information that they have. And I think if you look on the right hand side, you can see the COVID-19 outbreak. really starting in December, maybe a few stuttering events earlier, but basically starting in December. Maybe some of the local people in Wuhan were a bit slow to really put the red flag up, but I think, you know, once the Chinese got going, it was a pretty massive response, and they tried to lock the virus down, but you can see that in a relatively short order, it started to spread both within China and outside. By comparison, on the left of that graphic, or rather in the middle, you can see the SARS outbreak. There was more time during which to try to lock the virus down. It didn't quite have the spreading potential that COVID has, and that may be to do with the biology of where the virus is replicating. So on the very far left of this graphic, there's some summary figures from a very large study that was published using WHO resources to back up what was going on in China. And they described 72,000 cases, and the age distribution, the spectrum of disease, the case fatality, and so on. And also described the ability of this virus, just like with SARS-1, to spread in healthcare facilities. Next slide, please. And I just want to reflect on how close the Chinese came to being able to stop this one by public health measures. So, you know, we were all watching these graphs during January and February and thinking, oh, what is going to happen? You can see a big spike. Ryan, can you highlight about 12th of Feb somewhere? There's a big spike there. That was when they changed the case definition to include cases that were typical and had changes on computerized tomographic scans of lung, but were PCR negative. So that's a big spike there. But basically, we were watching that epidemic curve day by day, thinking, are they going to manage it? Are they going to lock it down? And as the days ticked by, we thought maybe they would. And we got towards the end of February, and then we heard that it was in northern Italy and it was in Iran. And we thought once it had got there, there was really going to be very little hope. And you can see the enormous wave that ensued, and it became essentially a European and then North American problem. And you know, the cat was out of the bag. So it was a very, very, you know, hanging on by a thread. I just so wish that we'd actually managed to lock it down in late February.
Peter, how
okay, so next,
do you think the first wave would have been if it had emerged in Europe or North America, say? How different would things be?
I'm afraid we would have found it very hard to lock down in the same way that China has. I mean, China has a very compliant population generally. They're pretty authoritarian the way that they can introduce measures. They're very used to wearing masks. I think it would be really hard for us to have managed to do anything better. I think it's inappropriate to say that the Chinese should have done something else. I think they did a really good job and just failed to make it. Yeah, next slide. Okay, so in terms of the disease spectrum that was described originally, so you know, I think we all need to remember that it is often very mild, and especially in younger people. So you know, I personally don't think it's so mild that I'm happy to support the use of this particular agent as a human challenge virus, even in younger people, because we have seen some very severe disease in young fit people. It's rare, but I just think it's clearly a very dangerous virus. So it's the ones that get the more severe forms of disease that obviously we're seeing in hospitals, you know, with respiratory failure, often not particularly breathless when you consider the very, very low oxygen levels that the patients are presenting with, particularly if they've just walked into the ward or walked into the A&E or walked upstairs. They can get really low oxygen saturations and don't seem to quite report the breathlessness that you might expect. But there's a very high rate of lung infiltrates, and sometimes it's hard to actually identify the virus on PCR, but maybe we'll get onto that a bit later. the lung infiltrates are often quite characteristic, and it's possible to diagnose it. So in terms of the time course, so there's a typical interval of about 5 days before exposure and the onset of real viral replication in a form that causes symptoms, and the symptoms tend to be mainly respiratory and to do with replication of the virus in the respiratory tract initially, although maybe more symptoms in the lower respiratory tract after the replication has really got underway in the upper respiratory tract and maybe the oropharynx, maybe even the oral cavity. Then once the disease is starting to progress in the first week or so, the viral load declines as measured by PCR, and the infectivity also declines. So after, particularly once the antibody response is cut in, which starts at about day 8, say day 7, day 8, and reaches a plateau, depending on the assay, sometime between 2 and 4 weeks after the onset. Once the antibody response is cutting in, then actually recovering infectious virus can be quite difficult. So next slide, I think, illustrates this. This is the time course of the clinical disease and is depicted in these colorful pictures just below the title of this paper. And you can see that the onset of fever and cough can be quite early and quite persistent, followed by shortness of breath when the pathology is mainly descending into the lungs, and then intensive care admission may ensue a day or two later. Some people are then going to need mechanical ventilation if their lungs fail, and at that point it becomes a really quite life-threatening condition. So the typical picture is that the disease lasts a couple of weeks, early viral phase, late immunopathogenic phase, and the characteristics in the peripheral blood are that the lymphocytes seem to be depleted from the peripheral blood, probably because they're going into the lungs. And with that, there's a rise in various markers in the blood. So IL-6 is one of these. You can see that in that middle panel. And that can increase over time in those that are getting more serious disease, which are the ones in red. So those are non-survivors in red. You can see that going up. Ferritin, a marker of inflammation. LDH, just below that, is a marker of tissue damage. So that's, again, a bad marker to see elevated. Up at the top, we've got D-dimer. Come back to that. That's a sort of indication of active thrombotic activation, which can be intravascular, and again is a bad marker. Let's go on to the next slide. Okay, so we were mentioning about the disconnect between the PCR detection and the infectivity. This is showing the PCR positivity over time in a set of people studied in New York, and you can see that you can remain PCR positive for quite a long time after the symptoms have resolved. Now, it always irritates me in publications or in reports when people say, you know, this is viral shedding. It sort of implies that it's infectious. But if we go on to the next slide, this is a graphic from a now classic paper, which was from southern Germany, when somebody came from to visit a factory in Germany from Wuhan and brought with her the virus and had a series of business meetings and ended up infecting a number of people who were previously fit and well. And you can see in the graphic on the left, there's a regular sort of decline in virus load measured by PCR, both in throat swabs and nasopharyngeal swabs. But if you look on the right, you can see in panel B, seroconversion really cuts in after about 7 days and then plateaus using this assay by about 14 days. And that once the virus-specific antibody has started to accumulate, then cultures become negative. So instead of having positive cultures, the cultures are all negative after about day 8. So at that time, You can still detect the virus by PCR, but it's not infectious. Okay, next. So on to ISARIC. So this is the great legacy from all the investment that we made in preparedness for pandemics back in 2009 when we had the flu pandemic, and where we really managed only to respond quite slowly. So ISARIC was set up as a preparatory study that would be ready to go if there was another outbreak. And a bit of a thankless task trying to organize a study that's there in case something happens in the future, because you have to invest a lot of time and energy in putting all the protocols in place and getting agreements from all the hospitals to provide you with cases and so on, when you don't know if there's even going to be an outbreak. So, you know, hats off to those who really invested heavily in setting ISARIC up, including the Wellcome Trust and Callum Semple and others. So next. So this, because we were ready to go, the ISARIC 4C consortium launched immediately that we started to get cases. And I vividly remember, you know, late night phone calls trying to ensure that we could actually recruit those very, very early cases when we were starting to see COVID positive people in the UK, just in the first week. And we've now ended up with 43,943 cases enrolled in terms of clinical data. Of those, we've got some biological samples just at the beginning of the disease, which are called Tier 1 in 537, and 1,361 in Tier 2. So this is serial biological samples. So it's a massive study, the largest, I think, in the world, that's managed to collect this sort of data and also samples. So next is, yeah, so this is published within the past week, the first report from the ISARIC 4C, really just describing the first 20,000 cases or so. So we collected about a third of all the confirmed COVID cases in the UK from 208 hospitals, and 2,648 people contributed to this collection. It's just amazing to see what a national effort this has been, and you know, I pay tribute to all of those who did that at some personal risk, collecting all those samples, all that data which has gone into this consortium. So Callum is in Liverpool, Kenny is in Edinburgh, and Callum is running the LIMS system, which archives all the samples, and Anne-Marie was the first author on this first description. Next. This is showing some snapshots from that. So you can see on the left that there's a predisposition of chronic cardiac or pulmonary disease and also diabetes. I mean, high blood pressure doesn't come out quite so strongly. We're not quite sure. We don't have very fine granularity on the clinical data so far, but this is the sort of headlines that you can see. So a lot of people present with shortness of breath and fever and cough, but sometimes they present with one or two of those three. Next. Okay, so this is breaking that down a bit further. So in terms of symptoms, so cough comes top, fever second, shortness of breath third, and then you've got constellation of other symptoms running all the way down to things like conjunctivitis. We didn't actually ask if they were anosmic, but on other surveys, anosmia seems to be, if it's sudden onset, very distinct, very typical, can be an early sign, frequently present. In terms of predisposing comorbidities, so asthma is listed there, but not particularly at a high rate. It's probably represents roughly the rate that you would see out in the community. So there are various other chronic diseases that do predispose. I'd say we've got malignancy there, just for people who are worried about whether they might be immunosuppressed because of past malignancy. Quite a lot of those people now who have recovered from cancer therapy, if you're more than 5 years on, it looks like your risk is back to normal. So next slide. Okay, so this is showing some of the symptom clusters, and I don't know if you can read those on the screen, but there is a very distinct cluster, sort of top right. I know. Let's get a start in the middle. This is with the big one in the middle. That's it. So big one in the middle. This is wheeze, cough, sputum, chest pain, and shortness of breath. Okay, so that's what you'd sort of expect, but there is a diarrhea and vomiting cluster, sort of a tighter little box just below. So remember that these coronaviruses do tend to have dual mucosal tropism, both for the lung and also the down, and then conjunctivitis and bleeding and so on more rarely. Next slide. Okay, so this is showing the very strong age susceptibility, and you can see that those of you who are sort of in your 30s and 40s really have very low risk, and the people who really need to worry are the people who are over 50, over 60, and certainly in your 80s, very high severity. Next. And this shows the survival curves of different age groups. And I think it's pretty sobering for some of us to see that in the age 50 to 74, the survival curve is pretty dismal. Okay, next. And this is a graph that wasn't in the publication, but which has come out of this massive study showing that obesity is a risk factor, but particularly if you're sort of middle-aged. By the time you're elderly, that obesity signal disappears, perhaps because people who are very overweight may have died early. Next slide. Okay, so I think, you know, this has been a wonderful experience to see this come out so quickly. And one of the reasons for emphasizing it to you is that we very much like further requests for collaboration on analysis of samples or data. So I'd like to hand over to Ryan to talk a bit about research going on and the research, you know, the roadmap for research priorities, which came out as this recent report from the Academy of Medical Sciences and the BSI. So over to you, Ryan.
Great, thanks. So the title of the talk was about controversies within COVID-19, and really the aim of the last 10 minutes or so is to highlight a couple of areas of active research where we really need to get a good understanding of what's going on in COVID cases. And I'm going to highlight two areas particularly. One is the ability to have reinfection. So that is based on, or similar to the first question that you had in the polls at the start, where I think 51% of you said that you thought you could have reinfection. And I'll talk through some of the data that exists that suggests whether or not that could be the case. And also about immunopathogenesis, so whether the immune system is harmful or beneficial in COVID-19. So the first of these that I'll talk about is cross-protection or enhancement of disease based on immunity. So really, after you've had the virus once, will you be protected or will you potentially have enhancement of disease? So the first piece of the kind of immunity puzzle really is antibody generation. It's the first thing we look for in protective immunity. So Peter outlined a lot of this research earlier on, but really we think that after about 14 days of symptoms, your antibody titers plateau in the blood, so you get a strong antibody response, and that peaks around day 14. The size of this antibody response seems to be positively correlated with age and inversely correlated with lymphocyte counts during acute disease. So combined, older people with lower lymphocyte counts have a greater antibody response. Those two factors, both age and low lymphocyte counts, are also associated with disease severity. So this paper suggested really that maybe there was an association between severity and the scale, at least, of the antibody response. And this preprint that came out just over a week ago, I think, on bioRxiv, suggests that this may be the case. So in a relatively small number of cases, they've looked at the amount of IgA in the respiratory tract and the amount of IgG in the serum in these patients over time. And you see that in mild cases, in these two left-hand boxes, you have really quite transient increases in nasal IgA and serum IgG. And these are quite low-level responses. Remember, these patients had quite mild disease. Whereas in the more severe cases, cases that were hospitalized or that developed respiratory distress syndrome, they had far greater peripheral IgG responses, so much bigger antibody response in the blood, and also a larger but more diverse IgA response in the respiratory tract. And we think nasal IgA is particularly important for the ability to be reinfected with other respiratory viruses, and that may be the case as well with SARS-2. But this really important piece came out again very recently from Florian Kramer's group in Mount Sinai in New York, which showed that serological testing for past infection really seems very good. So in people who have PCR confirmed COVID-19, only 3 out of their cohort, about 0.5%, tested negative for antibodies in convalescence. So really, if you have antibodies and you're using the right assay, it looks like you've had SARS-2 in the past. Again, and in COVID-19 suspected donors, so those that didn't have PCR swabs, about 1/3 of these had antibodies, suggesting that there is an amount of people who test negative by PCR, but still produce antibodies at some point later in convalescence. So first question really is whether those antibodies are universally beneficial. Will having antibodies circulating in your blood protect you from reinfection? That's pretty certainly the case for most respiratory viruses, but there is the potential for antibody-dependent enhancement of infections. And this is a study using the MERS virus, so not SARS-CoV-2, but there are experiments or assays going on for antibody-dependent enhancement with CoV-2. But this assay looked at the ability of MERS virus to enter HEC293 T cells in vitro, so in tissue culture, in the presence of neutralizing antibody at different concentrations. And they find that the ability of MERS to infect these cells increases here if the cells express the Fc gamma receptor 2. So this suggests that there are certain concentrations of antibody where the protective effect of neutralizing antibody is largely lost. There's a potential enhancement through uptake into Fc gamma receptor expressing cells. So this is in vitro work. But there are also studies looking at other coronaviruses in common cold circulation. So NL63 is a common cold coronavirus. And this study demonstrated that after a first infection with this virus, you have a relatively high TC value. So that's a relatively low amount of virus in the respiratory tract. And after some time, after about 80 or 90 days, if you become reinfected, you actually have a greater viral load upon reinfection with the same virus. This isn't due to genotype switching. There seems to be an effect that there is incomplete immunity to reinfection and actually potential enhancement of a secondary infection with this different coronavirus. So these things really need studying for coronavirus, for SARS-2. We need to keep a close eye on these. The other part of the kind of protective long-term immunity puzzle is the T cell response. So this paper came out last week in Cell from Alessandro Setti's group in La Jolla, where they've used class 1 and class 2 peptide megapools. So these are predicted peptides based on sequence alignment that they've used to look at the presence of CD4 and CD8 T cells in patients in convalescence from COVID-19. They found that 100% of participants who they took samples from had CD4 T cells circulating reactive to peptides from SARS-2. About 70% of those donors had CD8 T cells in circulation that were reactive to SARS-2. The scale of that CD4 response is also very closely associated with the antibody response as well. So this suggests that after you've had COVID-19, there are lots of T cells circulating that can react against these, split between CD4s and CD8s, and they're very closely associated with the antibody response. However, one of the really interesting things from this paper was that they looked at samples collected pre-COVID in unexposed participants. And they found that about 50% of these participants had T cells, CD4 T cells that were reactive against peptides from SARS-2. About 20% had CD8 T cells that were reactive. This suggests that potentially common cold circulating coronaviruses from historical infections have cross-reactivity to some of their peptides present in SARS-2. This cross-reactivity could mean that you have some degree of protection from infection with SARS-2. However, there's an interesting kind of dilemma in these data in that you're more likely to have severe disease in later life with SARS-2. You're also more likely to have had more exposure to these common cold coronaviruses. So this effect of potential cross-reactivity and cross-protection must be balanced against some of the other factors in COVID-19. So the second controversy that I want to talk about is the idea of a cytokine storm. And we often use cytokine storm to represent really an inflammatory response to immunity, to a pathogen. And it might be slightly misused in this context. And I'll try and explain why I think that. So when we talk about a cytokine storm, we really mean a dysregulated, exaggerated, pro-inflammatory response to infection, typically mediated by very high levels of NF-kappa B-driven cytokines like TNF and IL-6. As Peter mentioned earlier, IL-6 levels are higher in severe cases of COVID. This often mediates a multi-organ damage, particularly lung damage in respiratory infections, and damage outside of the primary site of infection as well, owing to a kind of systemic inflammatory response. However, when we look at the levels of inflammatory mediators in COVID patients, this paper looked at patients in ICU care, so really quite severe cases, compared to less severe cases, but still hospitalized. There were certainly elevated levels in these kind of more severe cases, and both were elevated above healthy control levels for both TNF, interferon gamma, and this list of cytokines here as well. However, there haven't been studies done head-to-head yet to compare these kinds of cytokine levels to real septic shock type cytokine storms that you'd see in toxic shock syndrome or bacterial sepsis. And if we were to compare this between the literature, then these levels seem considerably lower than you would see in those settings of real cytokine storms. So the cytokine levels are high during COVID infections, seem to be higher during severe infections, but don't necessarily translate to a cytokine storm per se. These results have led to the use of host-directed therapies in COVID-19. There are several host-directed therapies in trials, including IL-6 receptor antagonists and a few others as well. So I think a more accurate description of what's going on in COVID-19 wouldn't be really a cytokine storm, but potentially a viral sepsis response. So this is slightly more of a nuanced inflammatory response, really. And there are a few factors in COVID-19 that seem particularly specific. And one of those really is the formation of thromboses and blood clots, essentially, in the vasculature in these patients. So this could account for both lung damage and damage outside of the respiratory tract, including kidney injury and heart injury, which is quite common in really severe cases. So I'm going to take a few minutes just to explain the involvement of coagulation in COVID-19. And the first indication of this came from this plot shown earlier about D-dimer levels, which really are significantly, very unusually higher in cases of really severe COVID-19 compared to patients who have milder disease. D-dimer is one of the components of fibrinolysis. So it's a fibrin degradation product. Fibrin is degraded from a thrombus, which is a blood clot, essentially. So we can work backwards through these stages to look at what formed the blood clots in the first place, to then lead on to having this high level of fibrinolysis, potentially meaning that we could intervene much earlier to prevent these clot formations, which might inhibit some of the disease severity. And indeed, anticoagulation therapies are being considered for serious COVID-19 cases. And in about 50%, 50 or 60% of ICU admitted COVID-19 cases, we see these thrombotic events. So these are blood clots somewhere in the vasculature or in an organ, whether that's in the respiratory tract or elsewhere, that contribute to disease. And in fact, if you look at the post-mortem picture, then this shows that these microvascular thrombi about 9 times more common in fatal COVID-19 cases than in fatal influenza. So this really seems to be not just a feature of respiratory inflammation or even of fatal respiratory inflammation, but it seems to be quite an unusual and characteristic almost feature of COVID-19. That tells us something particular about how this virus might interact with the immune system, but it also tells us potentially about therapies that we can direct. So just in the last minute or so, I'll talk about how these clots might form. Firstly, we think that the infection drives damage to the airway, to the alveoli in the airway, in the kind of small air sacs in the deepest area of the lung. This damage results in the deposition of hyaline, which is seen in post-mortem, and damage to the endothelium, so that's the blood vessels lining the airway. This damage to the small vessels can cause hypoxemia, but also potentially causes these clots to travel around the body. And if you're interested in this area, then there's this fantastic review in Nature Reviews Immunology that's come out in the last few days that's looking at the potential mechanisms at the endothelium of endothelial involvement in severe COVID-19. The first of those is that the endothelial cells express very high levels of ACE2, which could mean that they are targets for direct viral killing. The viral-mediated downregulation of ACE2 could activate the bradykinin pathway, which is one mechanism for activating clot formation. But there's other things that are more familiar to immunologists. Things like inflammatory cytokines can drive endothelial cells to activate platelets and activate complement, both of which play into clot formation and kind of thrombogenesis. So there seems to be this interaction in severe cases with the immune system, the endothelial system, and really its clot formation, which is a feature of really severe case. So just to summarize, as our final slides, we think that the determinants of disease severity are certainly virally regulated. Viral load will have some influence in severity, as well as the sequence of virus. So there's relatively little demonstration yet that the virus is evolving in the human systems. Damage to the airway epithelium or even damage to the endothelium might encourage secondary bacterial infections, and particularly attracts lymphocytes and innate immune cells, which can contribute to immunopathogenesis. So hopefully now we'll return back to Doug for any of your questions.
That's brilliant. Thank you very much, Peter and Ryan, and apologies to everyone for popping up and invading virtually towards the end there. That was my virtual attempt of a chair standing up at the front of a conference, just reminding the presenters of time, but I didn't want to bring it to a close because it was such a wonderful presentation. We've had lots of questions. We'll have time to ask a couple, but before we get to that, we did want to rerun quickly the polls that we did at the beginning, just to see if your opinions had changed after what you've heard from both Peter and from Ryan. So just to run through those again, please do get your fingers ready. The first question is, do you think you could be infected twice with SARS-CoV-2? Do go ahead and vote on that one. And what we will do is we will close it there. And if we share the results of that, you can see that there was a vast majority here, 80% of you are saying yes, compared to 51% at the beginning of the presentation. So some real influence there, Peter and Ryan, in what you've said. And it'll be good to hear your thoughts on this in a second. And then if we go to the second poll, and we'll launch that, please do vote. So this is, which statement do you agree with most about COVID-19? And we will end it there. And if I share that result, first time round, it was 72% for the mostly beneficial and 24% for causes a lot of harm. And we're now 80% versus 19%. So a bit of a shift in opinion there. But again, it will be good to hear your views, Peter and Ryan, on that. So maybe if you could offer your reflections on pre and post poll, and then I can come to a couple of the burning questions that have been asked by our viewers.
Yeah, I do so hope that it's possible to induce a protective immune response. I'm sort of maintaining hopefulness, but I'm not sure. I just think, you know, it's going to be a very bad situation for us all if vaccines are not effective, or even if they cause an antibody-mediated enhancement or a cell-mediated immune enhancement, which we've seen a lot with RSV. I'd like to just, a lot of what we're learning about SARS-CoV-2 seems to actually reflect the sort of immunopathogenesis that we've been studying for years in RSV disease. So it's good to have put in all of those years studying RSV, I think, trying to understand this one.
Yeah, I think really it's just a word of caution that we aren't certain that having one infection means that you're protected from reinfection, and that we really need to understand that before we can have a really robust public policy around passport immunity or those kinds of things.
Mm-hmm.
Great, thank you both. So a couple of questions just to ask you. So there were lots of questions that had come in from our viewers, so apologies, we won't be able to ask all of them, but there were many questions around the lymphopenia that was seen in patients. And so I wonder if you could just expand on that slightly with questions around lymphopenia persisting in patients who died. Do you know what particular lymphocytes were affected? Are they traveling to the lung? Do they die off? If you could maybe provide a bit more insight around that, I'm sure our viewers will be delighted to hear.
I mean, I think years of studying respiratory viruses have shown us that what's in the peripheral blood can, very dynamic infection where you've got lots of turnover, often doesn't really reflect exactly what's happening in the tissue itself. And my personal take on this is that almost certainly those cells are being depleted out of the blood because they're taking a major role in the lung, in both defending against the virus, clearing up any virus which is present still after the innate immune response has done its bit, but also potentially contributing to immunopathogenesis.
Yeah, I think that some of the data suggests that perhaps CD8 cells are slightly more likely or slightly more depleted than CD4s, but really we don't know whether they are staying in the lung long term or whether they transit in and out of the lung. The data's not there yet, I would say.
Definitely. So work in progress, for sure. Another question came in a couple of times is to get your views on using convalescent plasma or hyperimmune preparations as a treatment for COVID-19. What do you think about that?
Yeah, shall I go with that one? I mean, it may well work. Yeah, and I think I mean it seems anecdotally to have been quite effective in SARS. I mean, the best time obviously to reduce the viral load would be early in the disease, but it might work even in people who've become hospitalized. It's well worth a trial. But again, I would say how important it is to actually do the trials. Lots of things that we think may be highly rational may actually be proven once you do the trial not to be effective. Just also to say with diseases like Kawasaki disease, polyclonal immunoglobulin can, in some way that's not quite understood, suppress the disease, even though it's not clear whether that's just due to a non-specific effective antibody or whether it's due to some specific antibody present in the polyclonal antibody that's given.
Yeah, and as we showed, really, the serological response in COVID-19 seems to happen quite quickly, really, within a couple of weeks at most of symptom development. So there's probably quite a narrow window of opportunity there where convalescent plasma might really work. But as Peter says, the trials are ongoing and need to complete, really.
Great. And final question, which is really coming back to the infrastructure that you both have helped develop and working on in terms of ISARIC. If the UK didn't have that in place, how long do you think it would have taken for the UK to start from scratch to get this up and running whilst also clinically managing the pandemic?
Ryan, do you want to go with that?
I think if we take the example of the 2009 flu pandemic, then it went through a more normal kind of protocol for funding. So the infection first arrived in the UK. There was then several months of negotiation before funding can happen. And that meant the whole first wave of the pandemic was missed, essentially. And it wasn't until the second winter of that pandemic when patients could really be recruited. That happens to be a relatively mild year. So there was a few hundred cases recruited in total for biological sampling. If you can compare that to now, we've recruited a third of all patients in the UK, which is quite phenomenal, and biological samples from about 1,800 of them in total. So, and serial samples from 1,200. So that's a completely different league to previous epidemics. It is a very different scenario. It's a very different infection, far more cases, for example. But it's entirely different, I'd say. It makes a really strong case for the need for preparedness for these events.
Yeah. So I think the swine flu was really a rehearsal, which taught us so much, and on which actually the response has now been based. So it's really good to see that we've got so much better at doing this now, and that we can mount this sort of response. It's fantastic.
Well, that's brilliant. Thank you very much both, and I'm pretty sure on behalf of the whole community and many patients across the world, we thank you too for your commitment and your dedication to ISARIC 4C and the other work that you're doing. You really are getting the insights that we need to understand this disease and find out as quickly as possible how we can overcome it. So thank you very much for that, and also thank you for your presentation, your time this afternoon. It is time, viewers, to draw this session to a close today, I'm afraid, but we will have the recording of this webinar online very soon, so feel free to view again and or promote to your colleagues far and wide as well, so that they get the benefit of Peter and Ryan's insights into COVID-19. And of course, there's a lot more that the British Society for Immunology is and can offer you as our members and the wider community. So please do keep an eye on Twitter, LinkedIn, Facebook. Please do visit our website hub, where we have some really useful resources, and read the emails that we sent you. You are our family. We are here to support you. So thank you everyone for joining. Hundreds of you joining live, which is fantastic. Thank you again, Peter and Ryan. Brilliant to hear from you this afternoon, and thank you to our supporters, Immunology and Clinical and Experimental Immunology as well, for making this possible. Sorry, Peter, did you?
I just wanted to thank you to you, Doug, and to the BSI for being such a big organisation. Great.
That's very kind. I'll give you that tenner later. Thank you, Peter. So everybody, we will see you next time. We actually have the next one scheduled already, Wednesday, the 3rd of June at 12:30. We will be hearing from Professor Paul Kleineman from the University of Oxford, looking at the antiviral responses that are happening within the infection and within COVID. So please do join us on the 3rd of June, 12:30, free to join as always, and it would be great to see you there. Thank you very much. See you all soon.